I'm Hakan Wallin. I'm from the Danish Institute of the National Research Center for the Working Environment. It's a sister institute in Copenhagen. And I will talk about high throughput method, toxicogenomics or transcriptomics. And there will be another talk today about that. And Peter Hoot will follow me and talk about proteomic changes in protein expression as a response to toluene, the isocyanate. And we will hear Dr. Schroeder talk about metabolomics, the changes induced by small molecules in tissues and cells and so on. So these are the major three techniques in high throughput global changes in, in comprehensive analysis of changes response to different agents. So transcriptomics or toxicogenomics, it's basically, you treat an animal or cells and you look for the changes. You compare with controls, just as regular toxicology examinations. You extract RNA from tissues or cells, and you produce cDNA. This you label with a dye. We used one we used in the same experiment. We used a control. Controls are mixed, but with another dye, so you have the same cDNA in the reaction, and you hybridize with a ship, which consists of an oligonucleotide with a specific sequence. Now, this ship, it covers the whole organism. In a human or in a mouse, it's about 30,000 different genes, and you have several probes for many of the genes. To treat all these this very huge amounts of data, you're dependent on computer capacity and experts in statistics and bioinformatics. And then you look at your data. You have to use some gene ontology to which is just identifying which is a gene. It's a kind of naming system where you have to sort things out. It's a moving body which is reorganized all the time to find out. Data mining, you go into the literature, you go into other people's experiment and compare your results. And I didn't mention, of course, you have to have a toxic endpoints if it's toxicogenomics. So we have looked at histology and inflammation, genotoxicity, other endpoints, which in the end then you correlate with the gene expression pattern. So I'm going to talk about the acute phase response. And what is that? That is a physiological response in the body to infection and trauma. And it's reflected by a rapid change in some serum proteins. This is plasma proteins, which is the definition more or less. You see a very dramatic increase in some proteins. You see here C-reactive protein, and I'm going to talk a lot about serum amyloid A. So these go up like many thousand times in response to an infection or another agent. Now, classically, CRP in particular has been of interest to epidemiologists because this is a predictor of cardiovascular disease, and it has been associated with air pollution. This is the traditional paradigm of the acute phase reaction in the organism, where you get the let's see if I can get the, where you get deposition of materials in the lung, you get an inflammation, a local inflammation. You get a release of interleukin cytokines into the circulation. Now this reaches, sorry, the liver. And the liver responds to this, and you get release of the acute phase proteins from the liver, which is the major site for production of CRP and other acute phase. So the focus is that you have classically thought, it's still, I think, the basic thought among most clinicians, that you get signaling via cytokines, which reaches the liver, and then you get an upregulation and a release of acute phase proteins into circulation from the liver. Now, yeah, here you can see there's been a lot of epidemiology, and you see here one example of how the plasma levels of serum amyloid A is a risk factor, is predicting the acute cardiovascular events in a human population. And there's many studies like this, most about CRP, but also about the serum amyloid A, which also is affected to a similar degree as CRP. Now, the serum amyloid A, we think is interesting because it's a part of, it's a pleiotropic agent. It has a lot of functions, but I'd like to focus here on its, that it affects cholesterol biosynthesis in the body. It's incorporated in HDL, and if you have a strong acute phase response, you will see the major protein in HDL, ApoE1, being replaced to a great extent by the serum amyloid. Macrophages that express SAA are more prone to engulf low-density lipoprotein and become foam cells. Let's go on. So, in mice, the patterns of the acute phase is a little different, but basically the same. So if you make a virus vector and introduce the SAA1 gene, there are 3 genes that are expressed in the mouse. This is a major one that's expressed from the liver, the hepatic one. So if you have overexpression in the mouse, you will see, now this is atherosclerotic lesions. So this is the basic mechanism for cardiovascular events. You will get atherosclerosis, you will get plaques in your vessels, blood vessels, and these contain cholesterol crystals and macrophages, and this can be measured in, normally mice don't get atherosclerosis like humans do. All of us get, most of us get some degree of atherosclerosis during our life. But this is a knockout mouse which develops atherosclerosis much like humans. And if you do overexpression of the SAA1 here, you will see that there is an increased lesion area of plaque. So there's also, as I mentioned, SAA induces the foam cell formation in macrophages. So the foam cells are the macrophages inside the plaques. And this is another measure this is an in vitro experiment to look at the uptake of cholesterol into macrophages. So we have done quite a lot of gene expression experiments. Especially, I would like to mention the Health Canada in Ottawa, where we have collaborated, and they have done all the RNA, the transcriptomics for us. So early on, we have actually looked at the axis of IL-6 and the expression of SAA in liver, and we were surprised to see very subtle or no effects in the liver. And we didn't really look at the lung, but when we did the global gene expression, we found that the serum amyloid A3, so this is the major species that expressed in the mouse outside the liver. This is the most changed gene in the lung. And you see 65-fold more expression on day 1, day 3, and it still remains some overexpression on day 28. These were mice that were treated, deposited carbon black nanoparticles in the lung. We also saw this with titanium dioxide. So this is an experiment with the deposition of titanium dioxide in lungs, and we saw the global gene expression. Again, the acute phase signaling is a major reaction you will see from particle deposition in the lung. Yeah. And we also looked at inhalation experiments, and we see the same type of, this was intratracheal deposition, most of the things I showed today. We use that as a preferred method. So we have had a lot of interest in carbon nanotubes. And this is an experiment with multi-walled carbon nanotubes, where we deposited by intratracheal deposition, 3 doses. And again, we see a strong upregulation, 1,000-fold and more of SA3 in the lung. And what's remarkable is that this reaction persists up to 28 days. We still see the reaction. And in this case, we also see a small response on the day 1 in the liver, but this is very marginal compared to the lung reaction. All right. So we also measured the protein in the lung lining fluid, the bulb fluid. We extract the inner part of the fluid in the lung, and we see an effect there. We have some sensitivity problems here. It's not so strong. I think the reaction is something about sensitivity rather than that it's so small. We also measured SAA in plasma, and we did this with carbon nanotubes, and we did it with other materials. And you see the reflection of the expression of RNA in the lung in protein in circulation. So this is a very important point to us. So when you deposit materials in the lung, you will see an upregulation of the acute phase in the lung, and you will see signaling of the acute phase in circulation. And this protein is much more stable, and I think the reaction is much greater than you would see from the cytokine. Now, the SAA is very strongly correlated with the inflammation in the lung. So this is a lot of different experiments with different mice, and they have got different types of materials, and it's different times. And you see there is a very good correlation between the number of neutrophils, which is kind of a cardinal sign of lung inflammation, and the SAA expression. So my first conclusion here is we were looking for an hepatic response. The acute phase occurs locally in the lung, and it signals in the tissues. And there is a direct correlation between the surface area of the most materials we look at and the neutrophil influx. The neutrophil influx correlates strongly with the SAA expression. And SAA is a predictor of cardiovascular events. So we think this is an example of what you can do with transcriptomics. You will see the whole spectrum. You will identify small changes or large changes, and you will be able to correlate that with a toxic insult. So particles deposited in the lung, they cause an inflammatory response, sometimes more persistent. You get the circulation of acute phase proteins. These affect the vessels, the blood vessels, and promote atherosclerosis progression. And this is a risk factor this is a major risk factor for cardiovascular disease. Okay, so I will talk about something else. I don't know what my time is. I have to check now. Where am I? 20 minutes. 10 minutes. Oh, okay, so we did we were interested to look at the performance of in vitro assays in comparison with mice. There's a lot of public pressure to reduce animal testing. I think it's not always realized how central animal testing is in risk assessment, but of course it's a good thing if we can reduce both the costs and the animal use. So in this case we used another carbon nanotube. It's the Mise, which is the only carbon nanotube that now has been classified as a carcinogen by the IARC in Lyon. This is a rather long one. It's a very fluffy and rather thick carbon nanotubes. And it is the carbon nanotubes that really has been tested around the world. The reason was that the Mise company donated many kilos of carbon nanotubes to researchers around the world about 10 years ago. So we tested this by dispersing them in saline with some BALF extract to get some of the natural detergents that are in the lung. And we gave them to a MML cells. This is a mouse lung epithelial cell line that was established by our colleagues in Canada. It's not a cancer cell line, but it's established from a normal lung, has many features of type 2 epithelial cells. So we tested around a large range of doses. We tried to be in the same range with a dose of microgram per surface area of the lung. So these are the experiments that were done by Transcriptomics, and we did some confirmation later with RT-PCR of a range of proteins to assure us that there was no significant response at the lower doses. I will not talk more about it. So this is a bioinformatics handling of the gene expression, where you classify things into different pathways. And you see in the upper panel the expression pattern in lungs, and the lower in the cells. And you will see that a lot of the same themes come back. They are differently oriented. However, if you go in and look into the individual genes, there is a tremendous difference. It doesn't look the same at all. Even if it's the same pathways, most of the genes that are upregulated in vivo are involved in cell signaling, cytokines, transmission, sorry, not transmission factor, but different cellular communication factors. Whereas if you go to the cells, oh, sorry, to the left here, you will see a different pattern from the right, that is tissue. And most of these genes are involved in transcription regulation. It's transcription factors and similar things. So there is a very marked difference in the gene expression pattern in vivo and in vitro. So, in conclusion, some genes, some common genes are observed, but just as often they are reversed effects on the expression. Similar biological processes are perturbed, but the underlying mechanism and the outcomes are vastly different. And in this case, I'd like to be humble here. In vitro and in vivo models are not comparable. Of course, other people have found the same, and some people have found more similarities. I think the general conclusion today would be to say that it's a tissue like the lung, which consists of so many different types of cells. You have to be very careful about the interactions between different cell types, and so you have to be very careful to really go for a full belief in what you read out of in vitro experiments. Okay, how much time do I have?
Oh, you still have 15 minutes. 15 minutes.
15?
Yeah.
Oh. I will be a little short. So, in international risk assessment, adverse outcome pathways has been a theme that has been brought up the last 10 years or so. This is an integrated tool. There's a proposed system for handling this by the OECD. It is about identifying not the toxic effect and the relationship with chemical, but the whole mode of action and all the steps in between here. You look at receptor-ligand interactions, steroid binding to the receptor, DNA binding by carcinogens, oxidation of proteins, and you have to see how this is related to the cellular response, gene activation, protein production, altered signaling, and protein depletion. And this you can relate to the organ responses, altered physiology, disrupted homeostasis, altered tissue development and function. And you combine the whole chain into the mode of action, and anchoring here is a very important step in this procedure, where you identify this key initiating event. For instance, android receptor binding of different chemicals. You can, in vitro, predict the effects in the whole organism, or even in population responses. This model applies both for ecotoxicology and classical toxicology. And you need to anchor your initiating event into the toxic effect. So this is a very nice example. There are many, not many, but a few, where it has proposed to use transcriptomics in risk assessment, in quantitative risk assessment of benzoapyrene in drinking water. It was, you have to read the paper to get all the details. It's a long story here. But basically, they have done two approaches: a classical, looking at the exposure and the cancer outcome, and a new approach where transcriptomics in a series of animal experiments and the global gene expression pattern was used. And there was a selection of relevant endpoints that really are linked to a pathologic effect, cancer in this case. And they did a quantitative risk assessment based on the two approaches. And they are very, very close in doses, in the benchmark doses that produce the departure of baseline here. So this is an example of transcriptomics, and you can mention also metabolomics or proteomics, which can be used in risk assessment today. So back to my old slide, what is the use of these high-throughput methods, transcriptomics? First of all, I've demonstrated how you can achieve mechanistic information about the process of the toxicant. This great interest in Europe now is about the grouping of toxicants, in my field, nanomaterials. These are chemically very similar, but the physical structure or coating can dramatically change the biological activity, and we need instruments to be able to identify which physiochemical properties render a material more or less active. So this we call grouping. It's one of the new catchwords. So we can identify toxicity signatures for a pathological effect. I showed you that we can do in vivo, in vitro comparisons, and we can do cross-species extrapolation and see if this holds for humans, for different animals. It will be useful for identifying biomarkers that we can use for human monitoring of insult. And I showed you an example of dose-response characterization based on high-throughput methods. So if I have a few other people that have been involved in this work are from my lab, Ulla Fogel, Niklas Jakobsen, Anne Saber, Sarah Paulson will defend a thesis soon. And very important from Health Canada, Julie Bourdon, Sabina Halapanava, Manuel Hussein, Carol York, and other people. And pathology has been done by Alicia Mortensen, basically from the Technical University of Copenhagen. So, thank you.
Thank